Well, thanks, Usha, for that very kind introduction, and thank all of you for coming out today to hear about our research and to learn what's going on at Stanford. Really appreciate your interest in what we're doing. And so, as Usha said, my goal today is really just to illustrate how basic research, people on a quest to just understand how life works, really can contribute to things that everyone agrees are important for society and medicine. And so I'm going to pick one story to do that, and I'm just going to say at the outset that much of the research that I'm going to introduce you to is not from my own lab, it's from many other people, and I'll point out a couple places where we've made some contributions to the story I'm going to tell from our own work, but mainly it's a community effort that goes, dozens of scientists over many, many years. So Stanford is one of the best environments for biomedical research in the world. We have a long history of spectacular discoveries in all sorts of fields. A few of them are shown here on this timeline. So we can go back into the last century and think about the dawn of the age of molecular biology and the invention of recombinant DNA by scientists here at Stanford. More recently, scientists here have figured out how to control brain activity with light. There are a whole bunch of other ones listed, and then one here in the middle and from the mid-90s is one of our scientists, Matthew Scott, discovered a mutations in a particular gene that are important, that contribute to the most common skin cancer in humans. And so that's part of the story that I'll tell. So I'm going to introduce you to a biological signaling pathway called Hedgehog signaling, and so soon I'll come to the origin of the name and why it's important. And I guess one key takeaway is I hope that after we go through the story that you all understand why it is that we scientists work on things like fruit flies, which I did as a graduate student here, or zebrafish, which my lab works on now, and also many other people employ organisms like yeast and plants for their studies. And hope you see why we do that and also how they can advance medicine and also benefit everyone in society as well. Okay, so sort of the end of the story, which we're going to give away at the beginning here, is that FDA approved a new drug for patients suffering from an advanced form of skin cancer called basal cell carcinoma. This is what the drug looks like. Don't worry, there won't be an exam afterwards. Just want to show you that's the end product there of our story. This is a cartoon showing what happens when a person gets a basal cell carcinoma. So this would be normal skin. The pink cells are normal skin cells. These are cancer cells that are overgrowing. They're dividing when they shouldn't be, and now they're no longer respecting the skin barrier there, and so these can form a tumor in place, and then eventually they can spread to other locations and cause disease. Okay, so in 2012 then, the FDA approved a drug to treat one kind of skin cancer. And the same drug and derivatives of it are also being tested for possible applications in lots of other types of cancer as well. So really, we've only seen the beginning, I hope, of the clinical application of the work that began from a clinical standpoint with the approval of this drug. Okay, so what's the starting point? Well, it turns out that drug, shown in the previous slide, inhibits a biological pathway called Hedgehog signaling. Okay, so Hedgehog turns out to be one of the most important signals that allow cells in our bodies to communicate in embryogenesis, certainly, and on into adulthood. So why on earth would we name something Hedgehog, and how was it discovered? I guess the one key point is that so the story ends with a drug that is useful for patients that have skin cancer. But it did not begin with scientists saying, well, the goal of my research is to try to come up with a therapy for skin cancer. That's not at all how the story begins, right? The story begins with scientists on a quest to understand how embryos develop. So one of the great mysteries in biology is how does an animal emerge from a single cell, namely a fertilized egg, and turn into a fly or a fish or a mouse or a human. And so going back now to the 70s, and again, most of what I'm going to tell you about is not my work, but these slides illustrate some work from other scientists who are very well known and received the Nobel Prize in the 1990s for these studies, using genetic analyses in fruit flies. So they're looking for fruit fly mutations that have interesting disruptions in development. So one is shown here. This is work from Ed Lewis, one of the scientists who received the Nobel Prize in the 1990s. A normal fly and then a four-winged mutant variant, so really striking morphological abnormalities. And Ed realized that this would be a way to, this mutation might be a way to understand the genes that are important for actually determining whether the fly has 2 wings or 4, right? So how is it that the body plan is formed? Similar studies were going on in the lab of Christian Nusslein-Volhard and Eric Wieschaus, who also won the Nobel Prize together with Ed. They were looking at fruit fly embryos and larvae, so this shows the larval fruit fly here. And you can see that each of the body segments has this characteristic little band of tiny little hairs here that you see as the white spots. And they found lots of mutants, dozens of mutants that were turned out to be important. But one of them looked like this. And it looks kind of like a hedgehog, right? It's got hairs all over. It's a short little, cute embryo with these little spikes all over the place. So they named that hedgehog. And so the beginning of the story really is the discovery of a mutation that looks like that. And they didn't know anything about what the gene was or how the protein the gene encodes functions. They had no idea whether it would be relevant to cancer or really anything else at all. But their interest in it was understanding that mutations that look like this surely will tell us something about the genes that actually pattern the body plan, right? So again, how does the embryo develop? That was the initial question. Or how do different segments get specified to make wings versus different kinds of legs and things like that. Okay. So skipping a lot of work now, so again, dozens of labs, hundreds of scientists. It turns out that Hedgehog, that mutation that was found in the 70s, encodes a key signaling protein that would be made in one cell. So these are 2 cells here, and the little red dots are meant to depict hedgehog protein molecules being made in one place, traveling through the animal over some distance, and then interacting with what's called a receptor on a different cell here in the environment. So if we blow up this little interaction, we have a hedgehog signal on its receptor on the surface of this cell, and then there's a whole cascade of events that receptor hedgehog interaction triggers that eventually leads to changes in gene expression down here in the receiving cell. And so for scientists trying to understand how cells talk to each other and how embryos develop, these were fascinating studies. It was really work like this, and again, I'm just giving you a very high-level quick summary. And I'll say if you have questions, we'll have time for questions at the end, so feel free if you want more details to ask. But this work led to the discovery of a key signaling system that turns out to be important not only for fruit flies and not only in embryos and wings, but in virtually all animals and many, many different cell types, including in our own bodies. Okay, so hedgehog signaling started with this fly mutant, and it turns out to have led the scientific community to this amazing signaling pathway that controls many aspects of development. So I alluded to the fact already that this is a work of many people contributing to this over decades. It turns out scientists at Stanford have made key contributions to many different steps in the signaling pathway, and 4 of them are shown here. This is Matthew Scott, who discovered that mutations actually in the hedgehog receptor gene are the ones that contribute to that skin cancer. That's the focus of our story. So that was an important understanding, that a mutation in a hedgehog signal, or a receptor, I should say, is important, can predispose you for getting skin cancer. Phil Beachy is another prominent researcher in this field. He discovered that the hedgehog gene itself encodes this key signal, and he and his lab have done many of the experiments that lead to all the various arrows on those diagrams, understanding how the hedgehog signal is made, how it travels, how receiving cells act on it. And James Chen and Tony Oro initially were working with Phil and Matthew respectively, and they made key contributions then, and now they have their own labs here, so they're on the Stanford faculty and have been for many years. And they are also, continue to make key contributions in understanding how hedgehog signaling works. So again, many people contributed to the story. In our own lab, we had a small part in it. So this is cover of a journal in which we published one paper on hedgehog signaling in zebrafish. This is work from a former student of mine. I'm not going to go into great detail about our own work, but again, just to illustrate that it's many people over many years contributing to the understanding of how hedgehog works. So in this case, we were studying mutant zebrafish embryos, so not flies, but mutant fish, and I'll come back to mutant fish at the end, and we can talk about why, but it's a great system for mutational studies, just like fruit flies are. And Ian discovered that this particular mutation he was working on actually encodes a gene that's important for the hedgehog signal to be released from the sending cells and then travel so that it can be received by the responding cells. So this mutation then led us to make some important, to gain some important understanding of how the hedgehog signal is sent and how it moves to responding cells. And so again, each of the arrows in the diagram then might represent many papers like the one I just highlighted, the work of many different people working for years in many cases just to understand each little bit of the hedgehog signaling response. So I'm really summarizing a whole lot of work from many people in a short time. This was critical, though, to understand everything about how the pathway worked. And again, from a fundamental point of view, it was worth doing this because scientists recognized that hedgehog and everything about hedgehog must be important for how embryos develop, how cells talk to each other, and all these basic events that we've been striving to understand for decades. So that alone merits the really intense focus on hedgehog signaling. It turns out, though, that it really is clinically relevant, as we started at the beginning. One of the key advances, again, came from a paper from Phil Beachy's lab. And what this paper did was look at what happened to mice who have mutations in the hedgehog gene. And so this is a normal mouse embryo here, and this is a sibling that is lacking both copies of the normal hedgehog gene, so it's a hedgehog mutant mouse. And you can see that it's very abnormal. It has very severe birth defects, not shown here in this view, but they actually have just a single eye. Okay, and I'll come back to that in just a second. So very severe defects, severe brain defects. It turns out that these are rare, thankfully, but human mutations in hedgehog gene as well, and they lead to similar defects as shown here in the mouse. So too little hedgehog signaling. If you reduce hedgehog signaling, you get various sorts of birth defects shown here. Conversely, other labs showed that, including Matthew Scott, whom I mentioned earlier, that excess hedgehog signaling, the signal is sent in inappropriately high amounts, you get cancer of various types. So this shows a patient with many different nascent skin tumors here. Okay, so that's a patient with basal cell carcinoma, the condition that the drug was approved to treat. You can also get brain cancer. One example shown here. This is a tumor, and here's normal brain tissue over here, and other types of cancer as well. So too much hedgehog is clearly bad because it can cause cancer. Too little is also very bad because it leads to birth defects. And so all the arrows in the slide that I showed earlier about the signaling diagram really are, the purpose of those at one level is just to make sure that the signal is sent in exactly the right amount at the right time in the right place for everything to go normally. And then any perturbation to that has the potential to lead to either too little hedgehog, which would cause birth defects, or too much, which could cause cancer. Okay, so that's one key step, understanding that hedgehog isn't just a fly thing. It really does contribute to human diseases. Second, and now this goes back even before the mutational studies in flies to the mid-20th century. It had been, some farmers, or shepherds, I guess, raising sheep, noticed that some of their pregnant females were having babies that had a cyclopic condition, so shown here a single eye in the midline instead of the normal two. And other scientists showed that this condition was caused when pregnant female sheep ate this plant, which is the corn lily. And it was further shown that the toxic compound, the one causing this birth defect, structure shown here called cyclopamine, the compound that causes this defect is called cyclopamine. Okay, and so here's the structure. Now, I mentioned that work from Phil Beachy's lab and others had shown that when hedgehog signaling is disrupted, you get a phenotype something like this in a mouse. And so what, in this paper, what Phil Beachy's lab showed is that this compound, cyclopamine, inhibits hedgehog signaling. So if this compound is present during critical stages of development of the embryonic sheep, then birth defects result. And furthermore, it's known that those birth defects result from blocking hedgehog signaling in a specific way. Okay, and again, Phil Beachy's lab made key contributions to that understanding. Okay, so that also led to an interesting idea, again, another key paper from Phil Beachy's lab. If this compound cyclopamine inhibits hedgehog signaling, and also if too much hedgehog is what's causing cancer, and by then he knew that that was true from the work of many labs, then maybe one could use this compound cyclopamine or some derivative of it to actually block tumor growth, right? Because remember, too much hedgehog can cause cancer, so what happens if you block hedgehog signaling with this compound? And it turns out it has a really striking effect, and again, we're not going to go through the data in great detail, but this bar graph over here shows the extent of tumor growth in a control situation where there's no treatment. And then if instead you provide, and these are animal studies, cyclopamine, then you inhibit tumor growth, so you have essentially no growth over the course of treatment. And so that then, this paper here, provided key evidence that cyclopamine, or really in the end something like it, might be useful for inhibiting tumor growth. And so that then was a key insight, and it was really only at this point or around this time, so decades after the discovery of the compound causing the defects in the sheep and after the original discovery of the mutations in flies, that actually it became clear that there might be a way to harness hedgehog signaling, in this case to inhibit it, to bring about a therapeutic effect, namely helping patients with cancer. So just to summarize a couple key points then, it really does take many years of basic research to understand how something like the hedgehog pathway works and to figure out how it might be useful for a therapeutic effect, in this case, helping with cancer. Okay, so literally decades of work. The story started with work in model organisms, or in the case of sheep, with farm animals out there. No one had any inkling of where the story would end up when they were initiating these experiments. The idea was strictly that by using these innovative approaches to understand how development works, namely looking at fly mutants or fish mutants or mouse mutants or various other things, that we might understand something about life that is important to understand. But what we would discover and how we might apply it, no one had any idea at the outset of this research. So it really is just trying to advance knowledge, okay? But the fundamental understanding always leads to some sort of applicability, even if it's not clear exactly when or how or you know, what the use might be. It's important to understand that this is really the work of many people, so hundreds of scientists from dozens of labs have contributed to understanding how the hedgehog pathway works. From my own small part in this, the one paper that I showed, that work was all done by one former student in my lab, a graduate student at the time, working on his PhD, Ian Woods. And that paper and some others that Ian did when he was a student here launched his career, and now he's a tenured faculty member running his own lab. So this really does take a long time, and it's important to understand that the scientists making these contributions are at many levels, not just the famous lab heads, but everyone working there is contributing to the advances, and they're also gaining training that allows them to go off and be the next generation of leaders in the biomedical sciences. And so that's another key function of basic biomedical research. So not only to advance our understanding, but also to train the next generation of leaders. And so another key point to take away in the summary is that the translational phase of this research really only began 25 years or so after the initial work had been done to discover the pathway, namely those fly mutation studies that I talked about. And so it really does take a long time, and it's important that everyone, including scientists, you know, keep in the forefront of their mind how long it takes to really understand something so well that you could manipulate it and for a therapeutic effect without causing some unintended consequence. And so one takeaway, I guess, is when you hear in the news or, you know, read in the New York Times science section that scientists won a prize for discovering something about how flies develop, or in zebrafish or plants or yeast or any of the other organisms, you realize that number one, it really is important, and number two, even if at that time it's not clear exactly how it might be used, you know, there's a good chance that somehow that information will at least be a small part of a step towards a new cure. And so another point was that it actually, from my point of view, after the key proof of principle studies that I showed with cyclopamine and understanding that it could inhibit tumor growth, it didn't actually take that long to develop a drug that worked in patients. And I think, and of course there are many reasons for that, including the skill and dedication of the scientists at Genentech who were driving that. But I think another part is that those scientists were building on decades of work. So people had meticulously studied the hedgehog pathway for a long time. And so when it became clear that there may be a therapeutic application, they had a huge base of knowledge to build on and to decide how it is that we might inhibit the pathway to help patients with cancer. Okay, and then I just want to spin, so this is all work now sort of in the past, right, just to illustrate the power of basic research and give you some sense of the, the scope of the projects and the length of time it takes for them to evolve. I want to move more towards current research, and here I'm just going to pick one example of work that we're doing in our own lab. So we're using the same sort of approaches. We're finding mutations, in this case in zebrafish, and this is a picture of a couple of fish embryos here, and I'll play some movies in a minute, but that's a normal fish. And this is a mutant. And this is a living fish, and again, I'll play the movie in a second, but each of the green dots that you see here is a particular type of immune cell called a neutrophil, and we've got a transgene in there that labels each of the neutrophils in the whole fish green. And so you can see individual immune cells in the living fish embryo. The reason we can do that is that these animals are transparent. And so that allows us to watch cells move and divide and to see how they interact with each other or maybe fight off an infection. So one of the reasons we study animals like zebrafish in the lab is that they're great for genetic studies. We can find rare mutants. But another key advantage is that the animals are transparent, and that allows us to understand development or how immune cells move in a way that you just really can't appreciate by any method other than watching directly. And so I'll play this movie now. The computer cooperates. So there we go. So what you see in the normal case is cells kind of crawling around. This is a normal healthy fish. And they're just wandering around and throughout the body of the animal sort of looking for trouble, looking for any signs of infection. And every now and then you see one of these green cells sort of zip by. Those are the immune cells in circulation. And because of the way we make the movies, they're only caught in one frame of the movie, so they sort of flash on and flash off because they're moving rapidly in circulation. So we were interested in mutations that might tell us something about how the immune system interacts with the brain. And we found this mutation here, and this is actually a sibling of this one. They were raised side by side in the same little dish of water. And so it's basically the same experiment, and the only difference between that and that is that this one has a mutation that we found. And you can already tell before I play the movies that something is different here. If you were really studying this carefully, you would see that there are very few of these immune cells in the brain of the normal fish. And even before I start playing the movie, so this is the eye of the fish and here's the brain, you can see these immune cells present throughout the brain. Okay, and I'll play the movie now so you can convince yourself of that. And you'll also notice there are lots more in circulation. So you see many of them zipping by and a whole lot more that have invaded the brain. And so the idea then is that this mutation might control something about it might tell us something about the genetic control of the immune system. So in this mutant, there's this abnormal invasion of immune cells into the brain, and there's way more in circulation than there should be. And I'm not going to go through the details, but this mutation then led us to a new gene that keeps the immune system in check. So the idea is you do not want the immune system to just trigger some very active response unless you really need it, like if you're suffering from an infection. This is a case of a healthy fish where the switch has been turned inappropriately, so it has this abnormal immune invasion in the brain that leads to severe consequences for development. And we hope then that by studying this fish mutant and the gene that it led us to and other similar mutations, that we might learn more about the genes that control the immune system, and maybe even these studies will give us some new insights into disease conditions in which there's an immune attack on the brain, for example, or some of the unfortunate consequences of responding to a severe infection. Maybe we can use what we learn about this gene or some similar mutant, and we don't know whether it'll be this one or some other one, to first gain some important knowledge about the regulation of the immune system, and then second, we hope that this knowledge will lead to important steps towards new therapies for controlling the immune system when we don't want it to to be hyperactive. So again, we don't know whether it'll be this mutant or some other mutant, but the general approach is the one I illustrated with Hedgehog. We hope that by just wanting to understand how it is that the immune system is regulated, that we will, number one, understand how the immune system is regulated. We'll gain some knowledge that's important and interesting to science, but we're sure that some of this then can be harnessed to help develop important new steps towards new cures as well. So I'll stop there and take questions, but before I do, I just want to, again, thank you all for your interest in our research, and thank you for supporting Stanford, and thank you for joining us today, and I'll be happy to take questions if there are any. Thank you. Yeah?
How did you go about labeling being able to get the light emitting?
Right. Okay. So it's other scientists, particularly Marty Chalfie, who won the Nobel Prize for this. They realized that there is a protein that, in and of itself, is fluorescent, and it's called green fluorescent protein. So as you can tell, it's a green fluorescent protein, and it's very useful in all sorts of cell biological studies. You can express that in any cell you want and make the cell green, or you can even tag parts of cells with that and make the individual parts of the cell green. So what we did in this case was use a transgenic animal. So we used recombinant DNA to splice control sequences that express genes in neutrophils, the cells we want to look at, so that they express this green fluorescent protein only in those neutrophils. Then we put that DNA sequence into the genome of the fish, and then we can basically we label all the immune cells. And many people, we didn't develop this approach. Many people have used this in my lab and many others to understand, how not only how the immune system works, but we have transgenes that label particular cells in the brain, or a whole variety of other things, and to watch cells during regeneration after injury, and all sorts of things. So it's now a very commonly applied method. And again, though, it does depend on having a transparent animal. So that's a key step, right? If you did all this inside a mammal, you wouldn't be able to see anything, right? But in this fish embryo, we can. There's another question in the back there, yeah.
Yeah, so when scientists study mutants,
yes,
is the assumption that the normal organism is the best that organism can be? Could it be that the mutant is actually better than the original?
So you can find, it's a really interesting question. Yeah, we don't necessarily make any assumptions other than if you want to learn about how a process works, whatever it is, if you can find a mutation that alters it in some way, better or worse, that might be useful, right? So a lot of mutants do tend to sort of cause defects, like I showed the mutant mouse with the birth defects, for example. That's because there are a whole lot of ways to mess up a complex biological circuit like this. But there are some mutants that have striking phenotypes that can be advantageous in certain circumstances, right? And we found those too. I mean, some of the studies we do, and again, I won't go into the detail here for the whole crowd, but we have one mutation in one gene that might cause a defect, and then we look for another mutation in another gene that might reverse the defect, right? And so there are mutations under certain circumstances that can be beneficial, whether it's in a disease circumstance or maybe out there in the wild. In general, the ones that are truly beneficial are a lot less common than the ones that have negative consequences, but both do occur. Yeah, really interesting question. Yeah.
Yeah, I'm just curious, that kind of process you outlined that lasted 35 years from the first discoveries to the drug. Over time, has that process gotten faster, and what are the things that have made that faster?
Yeah, that's a really interesting question too. There's a lot of interest in trying to make it faster, and also especially, to me, even more important than fast, is having fewer failures at the end of the process, right? And I think there has been some progress for sure, like things that I did when I was a graduate student here now are archaic, and no one even knows what I'm talking about when I told them what I did on a day-to-day basis. They're like, what, you had to do that? And, they think that, like that we were working back before wheels were invented or something. Not quite that old. Although I was a teenager when the hedgehog mutant flies were discovered, right? So it really does take a while. So the short answer is there's a lot of interest in that. And there has been some progress because, just everything is, better, faster, cheaper. And then in general, though, what that allows us to do is more experiments. So it's not, so we can, every individual experiment might take a whole lot less time, but collectively now we want to do a whole lot more experiments before we really think we understand something. So whether the overall pace of, publishing papers or really making the big advances has accelerated, I think you could debate, but day to day, what we do in a day, what we can accomplish is much, much, much faster than in the old days. The other thing I would say on the point of time versus failure, there are a lot, and you all have read many examples, there are a lot of high-profile drug trials that you hear about in the news that end up testing a drug and showing that it doesn't really help with the condition. So I call that a failure. Right? And of course, in some cases, there are harmful side effects. That's really a failure. But these are cases where lots of time has been invested. They're super smart people with tons of resources, right? So these are multi-billion-dollar efforts by the time you get into a large clinical trial. So huge amounts of time and smart people's effort and money and patients, who might have had other therapies, all invested in these trials that fail at the end. And that's really unfortunate. So I think that's what we, really, if I could, wave a magic wand and say, what would you change? That would be it, to prevent those failures. And in my case, I have a bias as I'm a guy who works on fish for a living, so I'm a basic biologist. I think, well, that's really a shame, and there must be something fundamental about the way the drug works or about the nature of the disease or the process they were trying to modify that they just didn't understand. It's not that they're ignorant in the sense that, these are super smart people who have invested a whole lot into these trials, so I'm taking nothing away from the people who, push these projects forward. I have a lot of admiration for them. But still, an unacceptably high number fail. And to me, that means there's just something fundamental about what they were doing that they just did not know, and therefore they could not really have predicted the basis of the failure until after they did all this. And to me, the solution is not necessarily to try to go faster, but is to invest even more in the basic research at the early stages before you even contemplate, making a drug that's going to go in patients and spending that vast amount of money. So to me, it really is a call to invest more in basic understanding, even if it's not clear, which fish mutant is going to be the key one. If you, have a bunch of students in a bunch of labs studying interesting mutations, eventually you're going to find the one that matters. And you may not even know which one mattered until 10 or 15 years after the fact. But they'll all be important. And then some of them will be really important and maybe lead to a new trial or prevent a bad one, which is at least as important, right? So to me, it's not so much accelerating, but it's generating more knowledge and a deeper understanding before you take before you really commit to an expensive trial in patients.
How common are defective immune systems in humans, and what have been the most effective treatments to date and perhaps anticipated in the future?
Yeah, so great question, and again, I'm not a clinician, so there are other people out and around that you should ask that same question to, but in general, the hyperactive immune system is a major problem, so-called autoimmune diseases are a major cause of morbidity in the society. Multiple sclerosis, type 1 diabetes, a whole host of other conditions are caused by improper regulation of the immune system, so when it's attacking something in our own bodies that it shouldn't. And there has been, again, thankfully, a lot of progress in harnessing the immune system, so in this case, tamping it down to try to prevent some of the worst of those effects in some of those diseases. The problem, though, is that causes side effects. We all, of course, need our immune systems, and without our immune systems, we wouldn't survive very long at all because we're constantly under attack from microbes in the environment, and our bodies are constantly defeating those microbes, and we don't even know it. Right? So, the catch is, again, it's this question of balance, kind of like with hedgehog signaling. You have to have just the right amount of immune activity in just the right place and at just the right time, and sort of an appropriate level of response. And in many cases, the drugs that are used are very helpful, in the case of a severe, immune attack on some part of the body. But we don't have the perfect way to control the immune system at just the right level at just the right time. So a lot more work needs to be done to come up with better ways to modulate the immune system. And it would be great if there were ways to predict that something bad was going to happen and then prevent it from happening in the first place. That's the real goal, I think, and we're not there yet. But again, I hope that research going on at Stanford will contribute to that type of cure, that really sort of directed precision cure to maintain health rather than cure disease. Right? That would be even better. Is there another question? Yes. Maybe we'll take one or two more. But
so,
yeah.
So some fundamental science project, some fundamental science research that can help in the long run, maybe.
Yes.
But there are also research that can discover that research on the new drug that can cure people, that can make money immediately. So how does Stanford or any organization decide what's the funding that need to be invested and why? And
yeah, so that's a great question. I guess one key point is that these drugs that quickly help people and quickly lead to a lot of money for the companies that make them aren't so quick if you really consider, as we looked in the timeline here, all the background work that had to be developed often at great effort and painstaking dedication to understanding how something works. So really, those overnight success stories might be built on 35 years of basic research, as we saw with Hedgehog. And so that's one thing, is you can't have the quick score without putting in the time beforehand. In terms of allocating money, I guess my own bias is that basic research is really important. It also is not very expensive relative to these large clinical trials. And so a small project, just supporting one student's salary for a year, I had one student who contributed in our own small way to understanding how hedgehog signals are sent and travel. So just paying one student's, and he did every single experiment in that paper, right? So just paying his salary and then a little money for fish food in our case would have been enough to make that important discovery, right? And so that's a very small commitment of funding that not only led to the discovery, but also allowed that student to be trained as a scientist. So as far as I'm concerned, that is a phenomenal investment, and the return pays off not only in the initial discovery, but in everything that student discovers for the whole rest of his career and all the people he trains. So to me, that is a phenomenal investment. And obviously, we have to make the big investments in the clinical trials too. But when those go wrong, then that might be $1 billion plus that should have been, you don't know that until you do it, so I'm not trying to be a Monday morning quarterback, but you have, a lot of resources spent in the end unproductively, right? So to me, that's what you want to avoid. And so I would argue that if you're considering the balance between funding basic research and funding clinical, if in doubt, usually funding more basic is a good thing to do. Oh, maybe last question, and then
yeah, I was just wondering about your lab. How many students do you have in your lab, and do the students return year after year for several years? Just basically the structure.
Yeah, right. Who's actually doing these experiments? It's certainly not me. I can assure you that. Kind of a typical lab would be maybe a few graduate students, and then a few postgraduate students called postdoctoral fellows or postdoctoral scholars that are, basically getting training until they move on to some independent step in their career. So it could be half a dozen people, something like that. Some labs are bigger, maybe more like a dozen. A typical graduate student might be here for 5.5, 6 years doing a project like this. In the case of Ian, the one I highlighted here, he actually published 3 papers, and the other 2 were very different. So he was, very productive, but it's not unusual for Stanford students. And so over, 5 or 6 years, they gain independence. They become experts in their field. They publish original research, and then they move on typically to a shorter period of time called postdoctoral training, often about 3 years, and we have many of both here at Stanford. And then, then they go and work in industry or start a company or, start a lab in an academic institution like I did. And so the total amount of time, if you count, the undergraduate training, is about 15 years. But they're not just really training, they're becoming more and more independent. So they're also discovering, right? So by the time they finish, they are driving the projects forward. They are making the discoveries with their own hands that, are summarized in a talk like this. Right? Great. Okay. Well, thank you all very much.
Okay.